Hello everyone. In this class, we shall discuss about the eukaryotic transcription termination. We have understood in the previous video how exactly transcription initiation and elongation occurs in case of the eukaryotes. In this class, I will be explaining you about the transcription termination in case of the eukaryotes. Before I start, let me give a brief introduction or a quick look back towards the transcription initiation in case of the eukaryotes. In case of the eukaryotes, this is considered as the beginning of the gene. The gene has several regulatory elements including initiator sequences, Tata box, B recognition elements and several others. These sequences are essential for the initiation of transcription by RNA polymerases. In case of the bacteria, RNA polymerase itself can recognize the promoter, but when it comes to eukaryotes, there is no way that the RNA polymerase can initiate the transcription. It requires the assistance of several proteins called as transcription factors. These transcription factors binds to the regulatory elements that are present in the upstream of the gene and initiate transcription. So here the transcription factors that are required by the RNA polymerase 2 are designated as TF2 family transcription factors. Now, the first transcription factor that recognizes the Tata box of the gene is TF2D. TF2D has two types of subunits, TBP and TAFs. I have clearly explained about the function of these subunits in my previous video, that is in the transcription initiation in case of the eukaryotes. So TF2D binds to the Tata box, followed by the interaction of TF2A and 2B to the promoter, then TF2F along with RNA polymerase 2 is recruited to the promoter of the gene that has to be transcribed. Finally, TF2E and TF2H are recruited to this complex to form the pre-initiation complex. This is, we are calling it as pre-initiation complex. It is not initiation complex. Here, the difference is the tail of the RNA polymerase is not phosphorylated. The tail, in the sense, it is the C-terminal domain of the largest subunit of the eukaryotic RNA polymerase. This subunit has the repetitive sequence of YSPTSPS. This is amino acid sequence, heptapeptide sequence, which has several repeats, and this has serine residue. Every second and fifth serine residue is phosphorylated. This is already been explained in the other video. Now, the second and fifth serine residue, when it gets phosphorylated, it behaves as a binding site for several proteins. Now, this is very important to understand the transcription termination. So, as I said, the tail is phosphorylated. The phosphorylation is taken care by TF2H, the protein transcription factor 2H. This phosphorylates the tail. Once after the phosphorylation, TF2H also has helicase activity. Helicase activity means it opens up the double-stranded DNA and it forms the transcription bubble. Therefore, once after the opening up of the transcription bubble, the RNA polymerase can initiate the transcription. So, two sequences are essential. One is the phosphorylation, the other one is the formation of the transcription bubble. These two activities are taken care by the transcription factors including TF2H. Once after the transcription bubble is formed, you can see that the RNA polymerase is moving towards the gene and all the transcription initiation factors are left out in the promoter site itself. The RNA polymerase is moving simultaneously, it is transcribing the information that is present in the DNA strand in the form of the mRNA. The mRNA is hanging out of the transcription bubble. So during this process, capping takes place and some post-transcriptional modifications also takes place. This is for better information. I strongly suggest you to go through the video that explains about post-transcriptional modification to understand whatever I am going to explain about the transcription termination. Next, as I said, RNA polymerase is moving. It extends the length of the mRNA. As the length of the mRNA extends, so you can see that the mRNA is extending and there is a triangular shape. I am calling it as cap. Cap means it is 7-methyl guanosine biochemically. It protects the mRNA from 5' exonuclease activity. Also, capping is must if the mRNA should be recognized by the ribosomes during the translation. If the RNA is not capped, then it is not recognized by the translation machinery. So far, let's assume the RNA polymerase has reached the end of the gene. It has reached, this is the end of the gene. After, when it reaches the end of the gene, any process should come to the end. Likewise, transcription should come to the end. During this process, as I said, the tail of the RNA polymerase is phosphorylated and it carries several proteins. One of the protein that is carried is the set of proteins that are being carried are the proteins that are essential for the polyadenylation. Polyadenylation means here there is a protein called as CPSF, CSTF, cleavage factor 1, cleavage factor 2. CPSF means cleavage and polyadenylation specificity factor. CSTF means cleavage stimulation factor. CF1 is cleavage factor 1. CF2 is cleavage factor 2. So these proteins are carried by the tail of the RNA polymerase. When the mRNA is transcribed by the RNA polymerase, when the RNA polymerase reaches the end of the gene, it transcribes a specific sequence called as poly A site. This is the red color one which I indicated here is called as the poly A site. This has been transcribed by the RNA polymerase, and the proteins CPSF, CSTF that are being carried by the tail of the RNA polymerase recognizes these sequences, and immediately these proteins are transferred to the mRNA, specifically to the poly A site. This process is already explained in the post-transcriptional modifications video. Then what happens? What happens next? Here, as I said, these CF1, CF2 proteins cleaves the mRNA from the growing chain of mRNA. Like this, you can see the mRNA has been dissociated from the transcription bubble. It is bound by some other proteins. Let's not discuss about those proteins here. But what I am trying to tell you here is the growing mRNA has been detached from the transcription bubble. You can see here, this picture very clearly explains the mRNA has been detached, but very important thing that to be noticed here is the transcription bubble is not yet closed and RNA polymerase has not yet stopped its activity. how RNA polymerase is going to recognize the end of the gene and stops its activity. This plays a very crucial role. Otherwise, the RNA polymerase is going to continue its transcription activity further throughout the genome. It may do it. Therefore, I am calling this as the first RNA. First RNA is already detached. Since RNA polymerase has not stopped its activity, there is a formation of the second RNA. There is a formation of the second RNA. You may be wondered to know the second RNA that is being transcribed by the RNA polymerase can have thousands of nucleotides. That means RNA polymerase go longer way even after the detachment of first RNA by the CPSF, CSTF and other proteins. So RNA polymerase is continue to transcribe and it is producing the second RNA. Then every process should come to an end, as I said. Now, second RNA, it has no function. It may be biologically inactive because only the first mRNA is capped and it is from the beginning of the gene. But we don't know the function of this second RNA, which is unnecessarily being produced with thousands of nucleotides. To end this process, transcription should be terminated. There are several models to explain transcription termination. One of the most appreciated model is Torpedo model of termination. Here, the beauty of this model is, though the termination process is not clearly understood, you know that I already explained you, this is the second RNA that is being generated. The difference between the first mRNA and the second mRNA is, first mRNA had a distinct cap structure. As I explained you earlier, mRNA, as soon as it is formed, the capping enzyme guanylyl transferase adds a cap to the 5' end of the mRNA. But in this case, since it is a second mRNA, the capping enzyme has nothing to do with this, and the 5' end of the second mRNA has no cap. You can see that this mRNA has no cap. If this mRNA is not being protected, there is a protein in humans called as XRN2. It is a RNase. As you all know, the function of the RNase is to degrade the RNA. Now, I have indicated XRN in the form of the star. XRN recognizes the 5' end of the second RNA. Once after it recognizes, it has the high processiveness or high processivity. Once after the XRN recognizes this 5' end, it begins to chop off. It begins to chop off the second RNA. XRN2 degrades second RNA swiftly. Swiftly in the sense, it degrades the second RNA in faster than the RNA polymerase that is being adding the nucleotides. Therefore, it moves faster than the RNA polymerase towards the transcription bubble, and it is chopping off, or it is degrading the second RNA quickly. So the direction of the degradation is again 5' to 3' direction. And one more thing is, it is believed that this XRN2 RNase either pushes away the RNA polymerase out of the transcription bubble, or it pulls the mRNA out of the transcription bubble to terminate the transcription. though like this is a model that has been explained, and with the discovery of the XRN2 enzyme, there are still other models available, maybe allosteric model and some other models, but this one is most most people have accepted this model. So this is the way how the transcription is terminated in case of the eukaryotes. Yeah, as I have explained you, so this RNAs has moved towards the chopped off the degraded the second RNA, which led to the formation of the double helical structure and closing of the transcription bubble and release of the RNA polymerase from the template DNA in a way that it can initiate the transcription with a different gene. So this is all about the transcription termination in case of the eukaryotes. Thank you.